Welcome back to my channel. Today we are going to learn about one of the most important edible mushroom cultivated in tropical region, the milky mushroom, scientifically called as Calocybe indica. This mushroom is called milky because of its bright white color and thick milky texture. It is a tropical mushroom that grows well in the hot and humid climate of India. Unlike button mushroom that prefer cooler temperature. Milky mushroom cultivation has become very popular among farmers and entrepreneurs because it is easy to grow and it has highly nutritious and economically profitable. It was first developed as a commercial crop in India by scientists at TNAU, Tamil Nadu Agricultural University, Coimbatore. In this lesson, we will learn about the scientific classification of milky mushroom and its morphology and life cycle, the condition required for their growth, the steps is followed to cultivate and harvesting, and its nutritional and economical importance. By the end of the class, you will understand why milky mushroom is called the tropical queen of mushroom and how it contributes to sustainable agriculture and food security. This Calocybe indica milky mushroom is a very new species suitable for hot and humid climates, including tropical and subtropical region of the country, and can be cultivated throughout the year. It is a robust, fleshy, and milky white in color, umbrella-like mushroom. The optimum temperature required for their growth, it needs 25 to 38 degrees Celsius. It needs warm condition for their growth. Milky mushroom can be harvested 24 to 28 days after spawning. The total crop cycle takes about 45 to 50 days, and it have a shelf life of 5 to 7 days without refrigeration condition, which make them easy to handle, transport, and store the mushroom. An introduction of Calocybe indica. The cap size 10 to 14 cm in diameter. The gills are white in color. The stem is cylindrical and about 10 cm long. The mushroom has no ring on the stem and it has an oily smell. The spore print color is white and the spore measure 5.9 to 6.8 micrometer in length, 4.2 to 5.1 micrometer in width, and Calocybe is a very small genus that contain about only 40 species. The cap, smooth cap, and fleshy, milky white in color, and margin incurved when young and straight at maturity level. Flesh thick, firm, white, unchanging on cutting. The gills crowded, adnexed to adnate, so it's remain white, and stem it solid, stout, centrally attached. And texture firm and meaty and edible, highly nutritious. It is a saprophytic fungus, grows on paddy straw and any agricultural residue. And tolerate to high temperature and humidity. The name, the Calocybe, that is derived from the ancient Greek term kalos means pretty and cybe means head. And this is the scientific classification of Calocybe indica, and it needs the climatic requirements for their growth. And the temperature range 25 degrees Celsius to 40 degrees Celsius, and relative humidity 80 to 85%. Under low humidity, the young fruit bodies dry up or the upper surface of the mushroom become very rough. And light during fruiting time, it need low light, that means 200 kilolux is necessary for that growth. And ventilation during fruiting stage, it need more oxygen is required, and therefore bags are kept in well ventilated room. And material requirements, it needs substrate and use any cellulosic agricultural substrate such as paddy straw, wheat straw, barley straw, maize, jowar, and bajra straw. So these are the substrate we can use. And mushroom spawn, so it needs spawn and 3 weeks to 1 month old spawn. And organic supplements for improving productivity, one may use pasteurized maize meal, wheat bran, paddy hull, or boiled wheat grains, and polythene bags. And cultivation practice, it follows different stages: mushroom spawn production, substrate preparation, mushroom bed preparation and spawning, so casing, cropping, crop management, harvesting, and yield. Mushroom spawn making. The best substrate for spawn preparation are sorghum grains or wheat grains. These grains provide an ideal surface and nutrition for the growth of mushroom mycelium. The grains are half cooked in water for about 30 minutes. This softens the grains, making them suitable for mycelium growth without becoming sticky. After boiling of grains, the excess water is drained off and the grains are air dried slightly so that they are moisture but not wet. So proper dry provide it prevent clumping of grains and ensure good aeration inside the spawn bottle. Next, the grains are mixed thoroughly with 2% calcium carbonate. This step helps to adjust the pH and prevent the grains become sticking together. And the prepared grains are then sterilized to eliminate any contaminants like bacteria or unwanted fungi also. Sterilization ensures the clean environment for the mushroom mycelium to grow. So once sterilized, the grains are inoculated with a pure mycelium culture of Calocybe indica under aseptic condition. This transfer the fungal culture into the nutrient-rich grain media. And the inoculated bottles are incubated at the temperature 25 to 30 degrees Celsius. During this stage, the mycelium spread rapidly, covering all the grains. After complete colonization, the grains appear snow white in color, clean and healthy. This indicates good quality spawn, which helps to quick colonization of the substrate and result in higher mushroom yield. And second step, substrate preparation. Substrates serve as a nutrient base for mushroom growth and development. So experimental studies were tested with different substrate and results showed the wheat straw is the best substrate for maximum fruit body production. And substrate are chopped into small pieces for easier handling and faster colonization. And these are the substrate soaked in a water to improve moisture content is about 60 to 70%. And pasteurization or sterilization of the substrate that mainly remove the harmful microorganisms that prevent contamination. And next step, mushroom bed preparation and spawning. First, bag preparation. So substrate, use chopped paddy straw that it has 3 to 5 centimeter in length and soak the straw in clean water for 6 to 8 hours. And treat the straw in hot water treatment means 80 degrees Celsius for 1 hour or steam sterilization 60 to 65 degrees Celsius for 6 to 8 hours. Then removal of excess water and keep the moisture about 65 to 70%. And filling into the bag, use polypropylene bag or polythene bag. Fill with pasteurized straw layer by layer. And next process, spawning. 5 to 6% of grain spawn can use. So place one layer of straw and sprinkle spawn in the corner of the bag and repeat until the bag is filled. And finally sealing, tie the mouth of the bag with thread or rubber band. And incubation, keep this bag in dark room 30 to 35 degrees Celsius and humidity 70 to 80% and kept for 15 to 20 days until the full entire bag covered with white mycelium. Casing, it is the next process that after colonization or mycelial development in the bag, the mother bag is cut and make into two bags, two daughter bags, and press it the mycelial development. The casing is a thin layer 2 to 3 centimeter placed over fully colonized substrate to trigger fruiting. So after applying the casing layer, that is helps to transition from vegetative form to reproductive phase of mushroom mycelium. This casing soil pH should be very neutral to alkaline. It's around 7 to 8.5 pH for better result. And steam sterilized clay loam soil, the pH 8.4 that is produced maximum yield and more mushroom buttons. The casing material can contain soil, sand, peat, compost, farmyard manure or coyote pit waste. And it contains no nutrients. It maintains only the moisture level and regulates CO2 and support aeration. And microorganism in casein stimulate the formation of primordia, and that means make baby mushrooms. And ideal condition to develop the baby mushroom, that it need 28 to 30 degrees Celsius with a relative humidity 80 to 85% for uniform cropping of milky mushroom. And cropping. Cropping is the phase when mushroom shift from vegetative growth to reproduction. After applying casing, the pinhead will start in the top of the casing soil. A small white knots appear within 3 to 5 days after casing. These pinhead grow into a button or it become as a umbrella shaped fruit bodies. The ideal condition to development of the button, so it take 25 to 35 degrees Celsius temperature, 80 to 90% humidity. So fresh air ventilation is essential to lower the CO2 level and stimulate the fruiting of milky mushroom. So light for 10 to 12 hours daily, it support healthy fruit body formation. Mushroom develop in first flesh every 7 to 10 days, and cropping cycle lasts 40 to 50 days, producing the multiple harvesting. Next, crop management is very important because substrate is the main source of weed, mold and pathogen. So it must be prepared carefully. So substrate should be chopped and soaked away from the spawning and cropping area. Backfilling and spawning rooms are sprayed with 1% formaldehyde twice a week. During spawn running period, 0.5% formaldehyde is applied weekly, and malathion is used once a week to control the insect and pest. And cropping room should be secured with iron frames and nylon nets on window. And proper hygiene and pest control lead to high yield and disease-free crops. And harvesting of milky mushroom. The milky mushrooms are ready for harvest in 30 to 35th days after spawning. So depending on temperature and humidity of the cropping room. The ideal stage for harvesting in when the cap is still tight and slightly convex, that means button stage, that before it become flat. That harvesting should be done early in the morning or late evening to maintain the freshness. And mushrooms are carefully twisted and pulled out from the bag without leaving the shrub or prevent contamination. The base of the stalk is trimmed to remove adhering casein soil or substrate or mycelial mud. The harvested mushrooms are immediately collected to the clean baskets or trays and kept in a cool place, and delaying in harvesting cause cap opening, spore release, discolorization and reduce the shelf life. It gives the average yield 800 to 1000 grams per kg of dry substrate, depends on cultivation practice. And this is the small summary of cultivation of milky mushroom. So chopped paddy straw and the length 3 to 5 centimeters, soaked in cold water for 4 to 5 hours, drain out excess water. If we following the hot water treatment means soak these straws in 3 to 4 hours in hot water. If chemical treatment following means with carbendazim and formalin treated water can use. And drain off excess water, drying at 60 to 70% moisture level. Prepare the bed, incubate the bed for 12 to 13 days, and after mycelial development, cut the bag and apply the casing in the top of the bag, and bed can be incubator, regular sprayed with water and maintain the relative humidity, and pinhead appearance after 8 to 10 days after casing. So harvested after 3 to 5 days and packed and dried. It contains all the nutrients. It has 17.2% protein, 64 to 24% carbohydrate, 4.1% fat, 4% soluble sugar, 2.9% starch, 7.43% ash. It's a good source of vitamin B6, low in calories, fat and sodium, rich in crude fiber. It contains all the 12 essential amino acid: alanine, aspartic acid, glutamine, glutamic acid, lysine, valine, histidine, threonine, and tyrosine. Thank you.